while we're waiting for that, I guess I can give the kind of the more generic introduction to the seminar series and stuff. So first lecture of 2024, it's the UCLA CART Distinguished Lecture Series. Our series happens once a month with distinguished outside visitors. And from October through June on the Friday. It's sponsored by the UCLA Tarjan Center, the IDDRC, the Intellectual and Developmental Disability Research Center, the Brain Research Institute, the CanReach Training Program, and of course, UCLA CART, Center for Autism Research and Treatment. We'll take questions from the audience at the end of the talk, and there's a Q&A window in the Zoom for those of you who are on Zoom. You can ask questions there. We're recording the talk and we'll post it to our website as well. It's a free channel. We don't sell it through cable or anything like that. So it's really an amazing, great pleasure. I always, both personally and professionally, to have Mike Talkowski here today. I would say it's a little more than 10 years since he developed his independent lab. Let's call it a dozen years or so, dozen plus. He's become a leader in human population genetics with regard to structural variation and defining structural variation in the genome. And it's in the autism field where we kind of overlap quite a bit. And, his work has been, he's the leader. He runs the structural variation group. Everybody kind of defers to his judgment appropriately because he's really spent an enormous amount of time thinking deeply about structural variation in the genome and how it influences normal genetic variation and disease risk. And so he's led efforts to make maps of structural variation in the human genome across enormous parts of populations, kind of annotating, let's say, nomad in a way, with structural variation. Those types of studies that are of crucial importance to human genetics writ large. And again, in autism, he's really done the major work in structural variation over the last decade, leads all of the kind of consortium, autism sequencing consortium, all the consortium work there. I'll tell you a little bit. He trained at Penn State, majored in biology and psychology, and then went to do a postdoc, a PhD. He did his PhD with Bernie Devlin at Pittsburgh. So you might get that he's a Pennsylvania boy originally from the wilds of Pennsylvania. And then he went to Jim Gazella's lab at Harvard, where they were able to retain him there and keep him there. And he's really built an amazing program, and not just his lab. He's now the director of, I guess it's called he's the head of the Broad Structural Variation Group, which says a lot in and of itself. But he is the director of a genomics and technology core at MGH, but also he runs essentially what is a department in kind of he can explain it, medical genetics, molecular genetics at MGH. And so anyway, it's great to have him here. He's also a really easy person to talk with for students and a great mentor, and really and super collaborative, and everybody loves to work with him as well. So thanks for coming, Mike. I'm sorry it's not sunny here. I know it's okay.
Oh my gosh. I left 24 degrees and 20-mile-an-hour wind gusts to the 4-degree torture, or the 65-degree torture and 4-mile-an-hour winds that Dan's apologizing for. So I am, as Dan said, the director of the Center for Genomic Medicine at Mass General Hospital and Harvard Medical School, and we're split across MGH and the Broad Institute. I have been working in autism and genomic variation for quite some time. And I thought for this talk, we've done a lot of work in autism over the years. I'd like to try to give at least half a talk that isn't published, or at least as new results. So I will touch on the Autism Association work, but I really wanted to step back and take a bigger picture of where we're headed as a field and what's coming next. So ironically, I gave a talk at ACMG about a decade ago, and I think this is the last time I used the slide on the left. But I thought I would frame the discussion today in this way. So for a very long time, I have been working on sequencing genomes, going back to kind of the first technologies that we developed to do so and try to understand how the genome rearranges and how the chromosomes rearrange. And as things have developed over the years, the question really becomes about at what point are we really meant to profile a genome? How much information can we extract from that genome? And how much can we interpret about function, biology, disease onset, and disease prevention? And so in this study back in 2012, we sequenced the first fetus with whole genome sequencing to derive a diagnosis. It was a translocation that disrupted a gene, CHD7, and it was associated with CHARD syndrome and a number of phenotypes that resulted actually in early neonatal death. And the point was just the technology development could get us somewhere. It was an N of 1 study and not terribly satisfying, but it was kind of the next step. At the same time, well before it, and then over a period of time, this concept of cell-free DNA profiling, or the idea, and this has gone on in tumor biology, but that within a developing fetus, there are cells that apoptose, there is DNA released into the bloodstream, and if you profile cell-free DNA from the bloodstream, one can extract information about the fetal genome. And Dennis Lowe and Steve Quake and others pushed this forward in kind of 2008 to 2012 in a way that aneuploidy detection from cell-free DNA became sort of I would say the first real application of genome sequencing for precision medicine that had broad uptake. Why am I telling you this? I want to set the landscape for what we're going to talk about today in this grounding, that really genetics has this major influence throughout the developmental continuum and really across the lifespan, from the earliest periods of prenatal detection all the way through adult onset and late onset disorders. Now, what I'll talk about mostly today is this box of the disorders that really, the genetic variants that are most relevant to early development. And I think this is really writ large and very broadly applicable. So autism is something that, I know this community is very invested in, and I'll discuss it. But I think that the variation that we find, the principles that we'll try to frame the discussion around and the applications are very broad. And I won't get into it, but a lot of what we're doing in autism and fetal genomics actually have broad applicability to schizophrenia and bipolar disorder and ADHD and many, many other disorders. Okay, so how do we have this really insight into genetics that has been almost a revolution in the last, I would say, 5 to 10 years? It is this very simple fact that developmental disorders and neurodevelopmental variability overall can present with social communication and behavioral traits that contribute to reduced fertility. There is no way I'm telling this group anything new with that information, but I think it's always good to remind ourselves of exactly how evolution works on these variants. It very strongly selects against variants that contribute to some of these behavioral phenotypes in the population. And in schizophrenia and autism, we certainly see this as one of the strongest impacts on fertility ratio. When fertility ratio goes down, we're driving down variation that has impact on that fertility ratio into the rare variant spectrum. In doing that, we now have some insight into variants of high impact. And when we can find variants of high impact, we can begin to leverage those to understand what happens when we perturb the genome and what is associated with various diseases. Now, this is a study that we did for non-coding variation in autism. I'm not going to talk about it at all today. It's published. It was effectively a mission of many of us in 2016, 17, 18 to show that genome sequencing could be done, that we could try to advance the initiatives of genome sequencing into the cloud, that we could build infrastructure around that, and that we could define variation and interpret that variation. And it was certain to yield nothing, and it absolutely yielded nothing. And this is exactly why it yielded nothing, because if I have a random de novo non-coding variant in the genome, I have almost no way to interpret that variant. Even if that variant sits in an enhancer that I know is conserved, that I know regulates a gene, I don't know which base within that enhancer I can alter. I don't know what the code of that enhancer is because I don't have a triplet code cipher. So we know that already. and even moving into de novo variants and promoters or UTRs. On the other side of the spectrum, we have the very, very large CNVs that have been known in microarray studies for 20 years in autism. And if we just take any given de novo large CNV, you can see here a relative risk category that's about above 3. Now, it's not as strong as one may think, because in fact, these occur quite frequently as well, and we need to decipher how. But the effect sizes are strong, but they're very, very rare. As we move into de novo loss of function variants, it's actually, you know, I think that this is maybe not fully appreciated. Again, the effect size is just under 2 in autism when you have a de novo loss of function variant or perturb an individual gene. So it's not as dramatic an effect as one might think. And then all the other variation really is quite weak. So the goal here is to try to understand the genetics of that developmental continuum, to try to annotate the genome in a way that we can push these effect sizes out and move the noise of the benign variation into this range of the effect sizes, and try to interpret these variants. There are ways in which we can do this that we've been working on over the years to try to annotate the genome, annotate those variants, and then weight those variants that are most relevant for disease. So this is basically the overall conceptualization of what I often refer to as the genomic medicine cycle, or the idea that genomic medicine really begins with an individual and an individual's genome. It then has to advance to the population and all of the variation within the population. Only then, when we understand what's in an individual genome, what the variation landscape looks like in a population, and what's selected against in that population, can we really push forward disease association onto mechanism, onto translation, and then back to the individual again. We're engaged in all aspects of that cycle within our department as well as within my lab. I'm going to mostly focus just on the variation part and the barriers that we see right now. The first of which is complete ascertainment of genomic variation. And, you know, I think that everybody's aware of the telomere to telomere assembly and the idea that we can now assemble human genomes. And I have a whole slide deck on that I can show anybody at some point, but I think we're making real progress there. We need robust references, particularly for structural variation, as Dan mentioned, and I won't leave here without discussing structural variation a little bit. We need metrics for interpretation, and I think this is critically important. And we really need to understand variant to function biology, and I think V2F remains really the fundamental grand challenge in our field. We're doing a lot of work on this. I won't talk about it in this discussion, but I'm happy to discuss it today. Progress has been slow. I think while we are, you know, as a community and as a field, really driving many initiatives forward. You know, decoding the genome and its functional impact has been a major challenge. And then finally, pushing the boundaries of technologies coupled with sober benchmarking. And I think that's the one thing that I will try to convey here. We have new technologies, we have new methods, we have many things we can apply, and too often these new technologies, I think, are oversold as we're going to impact every patient if we can assemble them from telomere to telomere. And in fact, we can impact very few patients beyond what we can just impact with an exome, and I'll show you that. Yet there is much to be learned as we advance these technologies. And I think just a sober assessment of exactly where the technologies are taking us and what we can understand, it will help advance the field far more than kind of some of the larger statements that come out. Okay, reference resources. I think this is more descriptive, a few descriptive slides. I won't spend much time on this. I think at this point, many people understand the reference resources in the field. What I want to touch on is how they have been used to leverage these metrics of selective constraint. So the Genome Aggregation Database, or GNOMAD, started as the Exome Aggregation Consortium, or EXAC, by Mark Daly and Daniel MacArthur. We have since moved to this GNOMAD program that is led by Mark and Heidi Ream and myself and Ben Neal and Daniel MacArthur, where the idea is to bring together all of the data that we can in the community, produce site frequency spectra of variants, and then distribute those variants and those site frequency spectra to the community free of charge, no use requirements, use them as you like, and really use them for everything that you can. The outputs have been useful, certainly not only for just understanding site frequency spectra, but for understanding knockout identification, what genes are intolerant to depletion or loss of function, to power disease association studies. And I think really refining clinical interpretation has certainly been probably the most common use of GNOMAD. It's in most clinical variant interpretation pipelines at this point. It's led by a council and just amazing team of scientists and trainees. I'll just plug one thing, which is, a lot of the people in my lab that are doing this work, you start on a project that you're probably not going to finish for 3 or 4 years. We just released a version of Nomad that we started 3 years ago or 4 years ago. And you're going to, the minute you finish it, release it to the world without having done any analyses. What I'll show you right now, we haven't actually done the analyses or written the paper yet, and anybody can do the same thing. So this is sort of really, I think, for the trainees, a major investment of time and something that is very admirable. And it's paid off for them, of course, but I think it's really a great resource for the community. Okay, so here's the latest Nomad release. 730,000 exomes, about 25% are from non-European genetic ancestry groups. We have pushed very hard to try to introduce as much diversity as we can. We had the UK Biobank that we brought in, which was this blue wave of European ancestry, but we have pushed hard over the last couple of years to increase the diversity of Nomad, and there's a lot more diversity coming, particularly with this blended genome exome product that we're running in a lot of major consortia from underrepresented populations. It's not the variants themselves, though, that I think is as useful in Nomad as the metrics they produce. So I want to talk about loss of function intolerance, or this idea of constraint, and this conceptualization, loss of function observed over expected upper bound fraction, or LUF, which is an absolute impossible acronym that Mark Daly and Konrad Karszewski keep coming up with, impossible acronyms. But the idea is, in the population, certain genes tolerate loss of function. I think most people are probably familiar with this, but I want to set the grounding because it'll actually pervade almost everything I talk about. If we have enough data, we can set expectations for every base in the genome, what that mutation rate should be, and what we expect to observe given an effective population size. We can then look at the observed over expected variation in the population and start to predict exactly what genes, at least, and someday which nucleotides are intolerant to alteration. In doing this, we build a distribution of tolerance. And if we look by deciles, effectively everything that we have discovered for haploinsufficient genes and neurodevelopmental disorder genes live in the 3 most selected against deciles of LUF. So about 3,000 genes in the genome do not tolerate loss of function mutations. That's why a lot of the early discovery in autism, neurodevelopmental disorders, and fetal genomics have been the low-hanging fruit, which are the genes that didn't tolerate loss of function mutations. A lot more work has been done over the years in annotating missense variants. So here we're annotating genes. Here we're annotating individual variants. This is a score MPC from Caitlin Samoha and Mark Daly. We've moved on from this, but even at this stage, you can see that we can draw a line. And effectively, when we have individuals with neurodevelopmental disorders that harbor highly deleterious mutations, when we annotate variants on the end of that spectrum, very few controls actually end up with those variants. So we've done a lot more work, and we'll be ready to present it soon on kind of combining these metrics with alpha missense and with regional missense constraint in the protein, and ways to really dissect exactly where these variants are. We're not quite there yet, but even now you can see that these are really separating out variants. Okay, those are individual variants. We've built these metrics of intolerance, but of course, genetic variation is a continuum. And within every human genome, we have about 4 million single nucleotide variants, 400,000 and maybe 800,000 now indels per genome that we can capture. Quite interestingly, as we move to long reads, actually don't capture that many more SNVs, we capture more indels, but where the big blind spot in our field has been for a long time, and we're starting to close, is of course in structural variants. So reorganization of the genome and variants of greater than 50 base pairs is an arbitrary definition, but one that has sort of stuck. And these include not only copy number variants, but flips and swaps and rearrangements of the chromosomes and of the individual genes in the genome. And they turn out to be quite significant. So finding these variants has been a major challenge, from the first visualization of chromosomes in karyotyping to chromosome microarray to find very large CNVs, to now whole genome sequencing where we can get to individual nucleotide resolution. But there have been no individual methods that really allow us to read these like they do SNVs and indels. So we have built a lot of pipelines. I'm not going to talk about this at all, It's dry and it's technical, but just to say, over many years, we have now implemented a series of algorithms that we bring in together. We use long-read sequencing and validation data to adjust machine learning models to set filters and thresholds, and get structural variation call sets out of short-read whole genome sequencing. We have similar pipelines for long-read whole genome sequencing. They all live in Terra. They are in Google Cloud, and the idea is they're open source, openly accessible. Anybody can use them. They're complicated, but anybody can use them, and we're happy to do so. I think the more important point, though, is for this discussion is, what are we finding? So Nomad at this point is about 140,000 genomes overall that we've processed, 76,000 of which are consented for open release. This is what lives in the database. And we now have about 1.2 million structural variant sites. As you think about structural variation in the genome, I think this is the number that is worth keeping in mind. From short read sequencing, we've now pushed this to the point that the early studies were finding about 200 structural variants per genome. From just the short read genomes that a lot of you are generating right now, we can generate about 12,000 structural variants per genome. When we're not capturing those variants, we're missing really critical pieces of the genome. So I think that it is vital at this point to begin to integrate structural reorganization of the genome with individual base pair mutations and trying to understand exactly how these things interoperate. And this is one of the reasons why, is if we take all classes of common and rare structural variation and look at how many of them represent loss of function mutations, in fact, there are more deletions that disrupt genes than PTVs from SNVs and indels in common variant space. But in rare variant space, where a lot of you are thinking, about 25% of loss of function mutations that we can capture from short read sequencing, it'll go up a little bit from long read sequencing, are represented by structural variants. So it's an enormous amount of information that we're leaving out of a lot of the studies that we're doing. Now, I think at times this also gets oversold. It's not voodoo and it's not miraculous. These things live on haplotypes. They operate exactly as all other human genetic variation does. And in fact, more often than not, they're captured by linkage disequilibrium on those haplotypes. But when we think about fine mapping variants, or we think about how functional perturbations are occurring, understanding the rearrangement of the genome may actually be more informative than just understanding the tag SNP that is tagging a given haplotype, and we've shown that a number of times. Why else is it important? Well, turns out that all of our genomes are a little interesting. We all don't just have these gains and losses of DNA. We all are carrying around also these complex structural variants that we've been defining over the years, which started as sort of our first examples of chromosome shattering, which we thought were these outlier events that just happened in a few autism cases 10 years ago. And now we understand that there's many, many classes of complex rearrangements. So inversions flanked by deletions and duplications, or very large insertions, or in fact, multiple breakpoints in proximity to each other. And we've begun to define now all of these variants. 5 or 7 years ago, you might have seen one of these and interpreted that as pathogenic in the cohort you're looking at. Now we understand that they exist in all of us. And in fact, while they're selected against, and this is sort of a measure of their selection, the proportion of singletons, you can see it's actually quite high by comparison to other structural variants. They are abundant. So complex variation lives in all of us. And without mapping and defining those, it's very hard for us to interpret these genomes. There's also this extreme complexity. Now, these are very rare, but they're always fun to look at. Perfectly healthy adult 56-year-old in Nomad, we sequenced their genome. We found 49 breakpoints across 7 different chromosomes. The chromosomes, little pieces of the chromosomes were shattered. It turned out that it stayed relatively intact with a couple hundred kb gained or lost in various places, all shoved into a very small segment of chromosome 1. So if you assemble this person's chromosome 1, you'd start walking across pieces of chromosome, of all these different chromosomes in little individual bits. And yet it's all reorganized in a relatively balanced manner. And this person's walking around perfectly healthy. And, we had initially interpreted that as being relevant to cancer, being relevant to developmental disorders. And it turns out it's just natural variation in the population. And it turns out it's in fact persistent in the population. So in Nomad V3, we found this in Nomad V2, as we've now increased by about 10-fold, we find that this is actually polymorphic. This exact event actually lives in 3 people in Nomad. And we found other variants like this that are polymorphic. So it turns out that the genomes are amazingly dynamic, that we can tolerate enormous amounts of variation and really rearrangements that we never thought possible within our genomes and very often be perfectly healthy. What we've learned is that it's not what happens in the genome, it's what is disrupted in the genome. And so as the organization of many of these segments is rearranged, it's really the genes that are altered. And if those genes are tolerant to variation, then it has generally been the case that the proportion of singletons, they're selected against to some extent, but not necessarily associated with severe disease. Okay. So I told you about constraint of SNVs and indels. Now we have constraint against individual copy number variants within the human genome, and we can understand exactly how that works. Well, from copy number variants, if we look on the x-axis here, and we bin genes by constraint, and we look on the y-axis, and we look at the observed over expected ratio of structural variants, what we see is that deletions are highly correlated with protein truncating variant selection, not surprising. Turns out duplications are also fairly highly correlated with constraint against loss of function variation. And even these internal exonic duplications, which we haven't really thought about much, right? How often have you done a study where you've actually looked at 2 exons that are duplicated within a gene? Well, it turns out that's actually quite correlated with loss of function intolerance. So what we think is that constraint actually marks a general intolerance to all structural variation in the genome, and this concept of dosage sensitivity. So we have genes that we know don't tolerate mutation. The question is then, can we find the genes that don't tolerate gain and loss in the population, because those are the genes that we're really going to be missing. And just looking at time, I'm going to start to blow through some of these. This is published, and this is in the published section of the talk. So we need a lot of data to do this because structural variants are quite rare. As I said, the mutation rates are rare. So we started to aggregate all the data we could. And where does all the data on copy number variants live? Well, it lives in the microarrays that have been ongoing for 20 years. So over time, we were able to aggregate about 1 million individuals, copy number variation data from about 1 million individuals. We had HPO terms for many of them across 54 phenotypes, data from the PGC, data from a number of diagnostic laboratories, and then data that had been aggregated from academic centers over time. The goal was to find large segments associated with copy number variants as well as individual genes. I'm not going to talk about any of the segments or the gene work that has been done many times over the years. What I just want to mention is what we did with the data. And very much like Luff, the details is a whole lecture that is not worth giving. This is all published. But what we did is the exact same thing. We started looking at observed over expected ratios. And then we built a model where we used about 150 gene level metrics of the properties of the genes, where they are in the genome, what their constraint scores look like, what they've been associated with, what their indexes are, what information is there in mouse orthologs and other data, and built these dosage sensitivity metrics. So the concept is there are certain genes that are more intolerant to deletion, and they're haploinsufficient. There are certain genes that are more intolerant to duplication, and they're triplosensitive. And then, as I just showed you, more often than not, haploinsufficient genes are also dosage sensitive. When we started this, there were about 17 genes that the ACMG had annotated as triplosensitive and interpretable. I believe that was the number. It might have been a little less. Well, we predict, very much like we predicted with Luff and with haploinsufficiency, there's about 3,000 haploinsufficient genes, very similar to the PTV Luff rates, but maybe upwards of 1,500 genes that are actually intolerant to duplication in the genome that we haven't really incorporated in many of our studies in the past. And the effect sizes are certainly stronger overall if we look at broad phenotypes and deletion than duplication. But among the 1,500 triplosensitive genes, we have about 380 of them right now, and this is growing as we add more data, that are not loss of function constrained. So these are genes sensitive to duplication that in fact are not among the haploinsufficient genes in the genome, and yet have an equivalent odds ratio when we map them to developmental disorders and neuropsychiatric disorders. So this is really a class of genes that we have not interpreted for intolerance to gain of function, or at least to duplication and increased effect size, as we have for loss of function. And, if we start looking at scores and stratify by autism risk of mutations, and these are old data from, the old cohorts in autism, which I'll show you more now, but just trust me that there's a lot of data here on de novo variants. When we stratify haploinsufficiency and triplo sensitivity, we see enrichment of de novo mutations within autism probands for the more haploinsufficient and more triplo-sensitive genes. Not surprising. We, in fact, across all neurodevelopmental disorders, continue to see the same effects, and we see those effects whether we're looking at protein truncating variants or missense variants. But again, very much like those duplication-sensitive genes, the missense variants are not being fully extracted and annotated here. And here's an example of that. So if we took the 46,000 families with data, and I'll talk about these families and gene discovery in them, but for the dosage sensitivity metrics component, the important part is that we have de novo mutation from a lot of individuals with developmental disorders. We certainly have enrichment of PTVs in constrained genes, in haploinsufficient genes, that there's no surprise there. If we correct for known genes, the enrichment actually goes away. So these are very weak effects. In fact, it's not even significant against the permuted null. So it suggests that overall, most of the haploinsufficient genes in the genome were already beginning to get to a point where, writ large, the de novo variation for protein truncating variation is found. But when we look at the duplications, what we find, in fact, is the mutation, the enrichment of missense and PTVs in the duplication-sensitive genes is actually highly significant, and it remains highly significant after we remove the known genes. So there is a function of missense variation overlaying duplication-sensitive genes that are strongly enriched in autism, and we have not localized those to genes of interest yet. And I think this is an area ripe for further discovery. That closes sort of the resource end of everything and how we integrate this into gene discovery, which I'll move to. I just want to give one plug for the All of Us research program. For those that don't know, it's a national biobank effort. We have collected all of these data to date. The whole genome sequencing is moving forward. It's actually at 400,000 now. We are about to release in the next month or two structural variants from 100,000 individuals. SNVs and indels are already released from these individuals. And what we have been killing ourselves working on is these data. We've already generated 1,000 PacBio long read data sets from self-described African Americans in the All of Us data set. The data is up. It's totally accessible on the All of Us workbench right now. And what we're delivering soon, this year, I hope, in the next couple of months, it will be really highly curated variant call sets from genome assemblies of 1,000 individuals. The largest data sets we have right now is from our HGSVC cohort of about 70 individuals, and then the pangenome reference of about 100. So over the next several years, we're going to generate about 20,000 long read PacBio and ONT genomes and release those again to the community with complete T2T assembly done on them, we hope. Or that's the plan. Okay. So in the back end, I'm going to talk about autism, and then really where I think fetal genomics is going is how I want to close this. This is the autism group. So let's talk about the shared and distinct architectures of autism and neurodevelopmental disorders more broadly defined. And I have to say, overall, varied communities have come at this question in various ways. When I first started saying I wanted to invest in autism, a prominent member of the autism community asked me why. I'd really just be studying intellectual disability genes. And it stuck with me for a little bit. I wanted to push against it. Then over time, these debates have been happening. What component of autism is unique to autism? I will say that I think we're starting to make a crack in that. I think that we are beginning to try to understand some genes, pathways, and dimensions that are not uniquely autism. I'll never use that word, but seem to have different properties than individuals and cohorts ascertain for anything other than autism. And I'll tell you a little bit about that, some of which is published, some of which is not yet. These are the cohorts that I'm going to talk about here. At this point, we have 28,000 families, or 28,000 parents, and about 15,000 autism probands, as well as a case control study of about 5,000 more autism probands, about 20,000. We have added a very large number of individuals from Spark over the last year. We have also added a lot of data from some diagnostic companies. We have the mutation results. We haven't aggregated it all together, but in the next 6 months or so, I think that this will get a major, major boost in terms of discovery. And then I think everybody knows the deciphering developmental disorders, Kaplanus paper that had individuals ascertained for developmental delay, not necessarily for autism. There is about 20% to 25% of them do have an HPO term associated with autism, but they were ascertained for developmental delay, not autism. And then we have reference data from the UK Biobank. Okay, first of all, these are all exomes. I just told you how important copy number variation was in the genome, and yet that was all from whole genome sequencing. In the exome space, we have left copy number variants completely off our radar, and certainly in the diagnostic space, there is no question we can find copy number variants from exome sequencing data. We have developed new methods, a method called GATK GCNV. Again, sitting in Terra, there's a whittle there. Anybody can use it, and we're happy to help anybody generate CNVs. It's very, very cheap, and it costs a couple cents per sample. And it turns out that after several years of iteration, we can call CNVs about as well from exome sequencing as we can from genome sequencing at a resolution of about 3 exons. So anything greater than 2 exons, we can basically call a CNV, and at 90% sensitivity, it will match what we find in whole genome sequencing. Certainly anything that sits in an array and covers a gene, we can find from exome sequencing. And we have a lot of benchmarking data here. This is just the sensitivity and specificity from whole genome sequencing and exome sequencing at greater than 3 exons. And you can see that effectively when we call a variant de novo from exome sequencing at greater than 2 exons, it's always validated. The actual specificity was about 95%. So with these methods, we now can bring CNVs into play for exome sequencing in autism, but we're doing this, of course, across many, many different cohorts. And this is why. This is the impact. So I was showing you those variants that live at about 2.5 from microarrays. Well, when we get to gene level deletions in the first decile of Luff, we're now getting effect sizes of about 10 in autism probands compared to their siblings. And even with duplications, again, weaker, but effect size of about 5. So this is significant risk that we're finding in autism and neurodevelopmental disorders from copy number variants. And I think this is one of the things that I was really interested in. It's really our first glimpse into CNVs at the resolution of a given exon or gene in autism and neurodevelopmental disorders at any meaningful scale. So at this scale, I'm just going to walk you through this. These are the things that we've known for a very long time. These are odds ratios of de novo variants. We've known for a very long time about the segments of the genome or genomic disorders that live within these segments. And of course, they're the strongest effect variants in autism, neurodevelopmental disorders. These are the individual variants that we have all been classifying from exome sequencing. This is their relative frequency in unaffected siblings. And you can see PTVs and constrained genes, are an effect size right around 3. Well, these are the genomic disorder, all genomic disorder effect sizes. This is a deletion of a constrained gene, not in a genomic disorder. So in fact, as I started to hint at before, in many cases, it's not the genomic disorder, it's the constrained gene or the gene that's intolerant to mutation within the genomic disorder that is in fact driving that effect. And we can now find this at almost exon resolution, and the effect sizes are quite large. So deleting the constrained gene was actually stronger than just finding the genomic disorder itself, other than these ones that we have known to have very, very large effect sizes in autism. And the same thing for duplication. Okay, so with this information, we can now have these functional annotations. And this I'm showing you in autism, but this will really apply to any of the studies that you have ongoing within given cohorts. We have functional annotations, gene level measures of selection, and we can weight genes and variants for association. Bernie Devlin and Catherine Rader and colleagues within our Autism Sequencing Consortium have built this nice model that allows us to effectively weight every gene by a base factor based on its measure of selection and the observed mutation rates. And the point is we want to move to a space in which we're incorporating all information that we have. Copy number variants, deletions, duplications, protein truncating variants, as well as missense variants, and weighting all of those by the metrics that we understand in terms of constraint. And so that's what we've done. And this is, I think, an interesting result, and it's published, and anybody can go look up the details. But what I think we're starting to do in autism and fetal genomics as well is begin to define allelic architecture, begin to define genes that are associated with phenotype, and begin to define exactly the mechanisms by which they're associated. In some cases, deletions dominate. In other cases, rarely duplications dominate. Sometimes missense variants dominate. And for some genes, we know exactly where those missense variants live and the impact they have on the cell or on the neuron or on electrical activity. But overall, certainly protein truncating variants and de novo variants are still the strongest effect size by far in autism and neurodevelopmental disorders. Okay, so we have 72 genes from that study. This is going to get 3 or 4 times larger in short order in the next couple of months, or certainly by the end of this year as all the data is aggregated. But here's the insights that we've been able to gain when we combine those data sets together. So if you want to ask the question, not autism, but neurodevelopment broadly speaking, we can combine these data. We can do the exact same set of analyses. I'm going to blow through this quickly because I want to make sure I get to the fetal genomic stuff. We can use a series of methods to show that the association metrics work quite well. And we can permute these, and across studies, we find very, very high correlation between methods. So, it doesn't matter. Use an FDR, use a de novo test, use a Bonferroni, just do the statistics right, and you get the same results. There's a lot of debate in the statistical literature about that. But in this case, that's not the case. When we look at de novo mutations now, in developmental disorders, cases ascertained for developmental disorders, mutation rates and the impact of de novo mutations certainly much higher than in autism. This has been shown over and over again. We reproduce this quite significantly when we look in the genes most intolerant to variation, just like we had shown you before. So this is important in missense variants. Again, the same thing. If we annotate missense variants further beyond MPC, this number jumps to about 6 when we use alpha missense and other information, but that's about where we are. So in joint analysis, we now have about 400 genes that really meet genome-wide significant association with neurodevelopment broadly. So if one is trying to understand changes in neurodevelopment, these genes all have some impact, and at a very, very robust genome-wide significance level, about another 650 genes or so that are overall associated at an FDR 0.05. The CNV burden is absolutely dramatic. The odds ratio is 48.9 for copy number variants among these 373 genes. So it is really dramatic. Once we define the genes, and in particular the dosage-sensitive genes, they're almost never disrupted by CNVs. Okay. So the final note I will make on autism before I move on is this, that, I'm going to try to touch on this active debate of which I'm less invested in than others. I'm just trying to understand the genome and the genes and haven't built my career on defining phenotypes. But the idea is if we downsample that mutation burden that we showed was much higher in cases ascertained for developmental delay and make that match with autism, can we find genes or subsets of genes that seem to have higher mutation rates in a case ascertained for developmental delay or a case ascertained for autism? And the answer is yes. Overall, about 85% of the autism genes were estimated to be associated with genes associated with developmental delay, and about 70% of these genes are associated the other way. And of course, the power was always much higher in the developmental delay cohorts. So this isn't surprising. So overall, again, there's not this large number of genes that are uniquely associated with autism. They all have evidence for association with developmental delay. However, there are some genes, and don't use 36 and 82 because these numbers will change a lot over time. But the point is simply there are some genes in the genome that, whether you want to define autism in any number of ways, the fact is when we do studies where we ascertain for a phenotype of autism, there are some genes that have very different mutation rates than in developmental delay. And I will say in the analyses we've been doing where we have now gathered data from diagnostic laboratories where basically they have HPO terms or referrals, and they're meant to be the most severe cases. And I absolutely would have predicted we could not reproduce this effect. We very much do. So at some point, a diagnosis of autism is leading in a subset of genes to a much higher mutation rate in autism cases than developmental delay, broadly speaking, which is most of the genes and most of the effect size. So, are these autism-specific genes? No. Are they autism genes? No. But they are genes that have undeniably different mutation rates in cases ascertained for one phenotype versus another. And I think that speaks to the properties of autism, as well as the progress that's been made in phenotyping, frankly, to get to the point that from a genetic sense, we can understand this. Okay, so these are our analyses. We have about 650 genes associated at this point. They reproducibly cluster into pathways and cell types and have functional effects that I'm not going into today because, frankly, most of what we found here wasn't new from what we already knew. So we have not accessed new dimensions of functional pathways yet. We'll get there, but we're not there yet, to be honest. Okay, I think I only have about 5 minutes here. I do want to tell you the story, though. So, unbiased assessment. Where are we going next? I've told you we can define the genome. I've told you we can find genes associated with the genome. And I told you at the very beginning about this work we had done in fetal genomes. We did a benchmarking study to say, okay, as we move the whole genome sequencing, and we've done the same thing with long-read sequencing, although it's not ready yet, what do we find from a genome that we don't find from an exome, a microarray, or a karyotype, which have been the standard of care for a long time? We did this in those same autism cohorts that I just told you about with whole genome sequencing, and then we did the same analyses in a fetal cohort of 249 fetuses with a structural anomaly identified on ultrasound. These are, again, individuals that live in that very low fertility ratio I told you about. Their genetic architectures look very similar to autism. And I'm just going to cut to the chase because this is only relevant for the next part of the talk, which is that overall, if you had a microarray and you had an exome, almost all of the diagnostic variants are found. If you call CNVs against that exome, literally everything is found. Because as I told you at the very beginning, we have no idea how to interpret most of the non-coding genome. All we're adding is really structural variants that the genomes could grab that the exomes could not. But the increase in diagnostic yield in these autism quartets was only 0.4% between genome sequencing and an exome plus a CNV. Certainly, genome sequencing is superior to any given technology. But when you combine them together and have a karyotype plus an exome plus a microarray, it's about the same diagnostic yield. And I think that speaks more towards where we are in interpretation than where we are in genomics. Same thing with fetal anomalies, slight uptick in terms of what genome sequencing accesses just because of the variants, and there are a few more structural variants within that cohort. But basically, beyond aneuploidy, the power of sequencing interpretation is really living within the coding mutations in the genome right now today. And the reason I'm emphasizing this point is for this, that I think the next step is to be able to access that information as early as possible and to access that information within the coding regions of the genome so that we can begin to interpret everybody's genome. So this is the work that we're doing in the Fetal Genomics Consortium. And it's a consortium started by myself and Ron Wapner and Wendy Chung and others, trying to build cloud-based repositories for fetal genomics, the way that we have them for autism and neurodevelopmental disorders. We're working on stillbirth and structural anomalies. But it's this work that we've been doing over the last few years that I'm going to end with here, because I think it's interesting, and we just wrapped it up. So at this point, non-invasive prenatal testing is offered to mothers, to pregnant persons, in which you can do a screen for an aneuploidy. If an ultrasound is detected, then an invasive test is done, an amniocentesis or a CVS, in which one can do exome sequencing, genome sequencing, or microarray. There is a major push on newborn sequencing and getting to individual variation that may be relevant to early development. And then there's been all of these studies on rapid NICU sequencing. How quickly can we actually get the information to it replicate? Okay, great. How quickly can we get information when a neonate is in crisis and ends up in the NICU? And there's been work at Rady Children's Hospital and other places. You know, how quickly can we do this? Can we do this by nanopore? Can we do this by short read sequencing? But in fact, the major revolution in precision medicine has been the uptake up to 50% of pregnancies now that are getting NIPT. So the DNA from the circulating fetal genome is actually being accessed for aneuploidy and trisomy screens. and in some cases targeted CNVs, but maybe that information actually can be accessible to us so that we can capture everything I just told you about in autism from that fetal DNA. Okay, so I have 2 minutes, so I'm going to skip the details of the background and just get it to you. So the proof of principle, here's what we did. We did a maternal blood draw during pregnancies from the 1st to 3rd trimester of pregnancy. We extracted cell-free DNA, we developed an exome sequencing method, and then we developed a whole series of methods around analyses. And it's challenging because this is what the fetal fraction looks like. When you extract DNA, you end up with, in the 1st trimester pregnancy, about 10% of that DNA being from the fetus. By the 3rd trimester, it's much higher, but those invasive amniocentesis tests are done here in the 2nd trimester. So you go in, you do an amniocentesis, considerable risk to the fetus, considerable risk, not considerable, but meaningful risk, and it's a medical procedure to the mother. So at 200x coverage, what we were able to do is separate out all the alleles that exist in the mom and all the alleles that exist in the fetus from exome sequencing of that same Streck tube that was collected for trisomy screening. And just to orient you to what this means, well, here's the fetus, here's a mother that's heterozygous, here's the fetus that's heterozygous. So if you're heterozygous, half of your alleles are your alternate, half are the reference. Well, when you have a fetus with 10% fetal fraction, now it's half plus 10%. And so you can start to separate out these peaks for variants based on who the mother is, what the mother carries, what the fetus carries, and what the father carries. And of course, this is that money peak of those de novo variants that only exist in the fetus and don't exist in the mother. And what was really spectacular about this is it turned out that within that fetal DNA, just for time, I'm just going to say, if you don't filter by genotype and you just say, is the variant that's in the fetus when I do exome sequencing from a matched amniocentesis in that cell-free DNA? And it almost always is. Can we genotype it accurately? That gets a little harder, but we end up at about a variant sensitivity after genotyping of 90% to 92%, and before genotyping about 98 to 99%. So in doing so, I'm going to skip this and just show you this, that no matter when we collected the sample and no matter what the fetal fraction over time, those de novo variants and the variants that live in the father or are new to the fetus that we use in autism so strongly are always detectable. So at any period at which we collected the samples, we could define the de novo variants that live in the fetus and not in the mother. And that is really important because what it ended up allowing us to do is we matched standard of care amniocentesis in 14 samples that were referred for an ultrasound anomaly. And in 100% of them, we were able to find a concordant diagnosis with the standard of care invasive test. And that includes copy number variants, which we can talk about again. But the variants are individual splice mutations, copy number variants, carrier risk variants in the mother, risk variants in the fetus, as well as a number of other interpretable variants. And along the way, we also get a free carrier screen in the mom. So in fact, 60% of the pregnant persons had a reportable finding from carrier screening, all from this non-invasive test that's already being collected with a Streck tube. So this is where this work is going. And I really think there's a lot of impact for this. Again, not in the scary way of, oh my gosh, we're predicting polygenic risk and selecting fetuses, but in the way of right now, very much like that very first sample that I showed you, the first fetus that we sequenced, where a karyotype said there's a rearrangement, and it ended up in neonatal death due to Charge syndrome and CHD7 mutation. Had we sequenced it, we could have told the parent that this is the exact gene disrupted, not this chromosome's rearranged. The same example here, where instead of trisomy screening, we can simply give information that is useful and relevant. And we're already giving the families when we do amniocentesis or CVS, but we can give it to them earlier and we can give it to them as accurately without the invasive test. And it really suggests that we can take this information and interpret it throughout the lifespan. So when it's relevant in neonatal care, when it's relevant in adult onset care, this will already be in the medical record. There's a lot to do from day one to that interpretation, and we can't do any aspect of that right now. But as a proof of principle, this information is accessible, and I think this is really where the future will go. Okay, so I will leave it at that. I think I still left 5 or 10 minutes for questions. These are some of the people in my lab. My lab is awesome. They really They do an amazing job. We have collaborations all over the place. These are the nomad people that do all of that work, and really they get all the credit. I'm just the one talking about what they did. So thank you very much for having me out.
Well, we have, I'll handle the questions from the audience, but we also have some online, and Monica will ask those. Maybe we'll start with that.
I'm sure we have an online question. Is there any information on the cell type and brain region specific gene expression patterns of the autism specific genes versus DD genes?
It's such a good question. There's actually an entire section of the Fu et al. Paper on exactly this question. And you know, I am almost intimidated to answer this in a room with Dan in this community, but in fact, yes. And while the world has certainly tried to pursue this killer cell type for autism and developmental disorders, it's certainly not there. The reason I didn't talk about it is I don't think we found anything new. I think we found a number of the cell types enriched for constrained genes, enriched for genes associated with developmental delay, and enriched for genes associated with autism that followed very much in the work that Dan and many others had done over time, looking at co-expression of genes within various tissues. So broadly, there are a series of neurons and co-expressed genes that are enriched within those neurons. There's some minor differences between the developmental delay and autism cohort. And I mean, I can go into it in detail with folks if they'd like, but there was nothing striking within those enrichments, and it very much followed what had been published in the literature for a very long time, probably going all the way back to some of the earliest studies in brain span from here.
Yes, yeah, no, super interesting. It's a great question, Paimon. So,
Great question.
yeah, yeah, I was kind of struck by your take on, it's really great data too, from kind of whole exome plus, you know, the kind of clinical approach that one's going to take. So you know, right now, you know, it starts with microarray, or if there's a boy, Fragile X and microarray, and then if there's not anything there, whole exome sequencing, and you're showing like 7.4% yield and 7.8% yield in the other, and then with whole genome. And what Stan Nelson and others found here with whole genome, like, so it's part of the undiagnosed disease, and I'm sure you're aware of this because there's also a MacArthur, there's a Broad paper for about this too, is that it's true that the whole exome doesn't, the whole genome doesn't add that much because of this issue of non-coding variation. But what can help you there is having RNA sequencing to interpret that. And so I'm wondering, like, you know, so there are 3 or 4 papers now out showing that in different cohorts. The one from here, the Broad larger paper, there's one like Australian one. So I'm wondering, you know, kind of what you like from a clinical standpoint. Our approach thought is, you know, if we can, and they're negative with the usual whole exome plus, you know, microarray kind of stuff, then we'll go to whole genome plus RNA-seq. We won't do whole genome kind of alone. And you know, of course, if you have a muscle biopsy or you're doing a muscle disease, it's going to be much better. Half the genes aren't expressed in the blood that you're interested in, but still, I'm wondering what what your take is on
absolutely,
because you've looked at this.
We've looked at it extensively in many different ways. So no doubt the genome. So first of all, I'm a huge advocate that we should be running genomes as the costs come down, because you get everything that the other tests do, right? So there's no sense I mean, you know, if it were up to me, nobody would ever run another microarray forever because we can get all of that information from the genome plus. But where you are, let's say, genome negative, there has been, again, I feel like there's a paper or two, and then we hope that this will explain everything, and it just doesn't. There has been absolute useful yield coming out of RNA sequencing, particularly for when we get to the tissue of interest in the muscle biopsies and other things. For RNA-seq combined with the genome, this is very useful for missense variants, for splice variants, and in a small number of cases, I think regulatory variants, although we're a long way from that. There are examples, like we were talking about last night at dinner, where we did this study in the Philippines for a very, very rare lethal Mendelian disorder that has a later onset and is indigenous to one island in the Philippines. There, it was a structural variant that jumped into a completely intolerant gene. But it wasn't until we did the RNA-seq matching it that we actually could find that it was intron retention, alternative splicing, and just a 20% reduction in the gene expression. But the gene is so intolerant that that was enough to be lethal in later onset males. So there are a few cases where we would not have gotten anywhere without the RNA-seq. The challenge is 60, maybe 65% of the genes of interest are expressed in LCLs or fibroblasts.
Yeah.
Regulatory architecture looks different, regulation of those genes, and then even E-outlier analysis becomes somewhat complicated. So there's many challenges, but increased yield, no doubt.
Yeah, I mean, I guess like what I was like, how do you, like our thinking about it is if there's a 7% yield, you end up with like a 10% yield or something if you're running the RNA. Does that fit with your?
I think it has been very much phenotype driven. The Center for Mendelian Genetics at UDN has been doing a lot of this work, as you know. And I think where we have phenotypes where we can get to tissue, the yields I've seen have been, 5%, 7%, if it's the right set of genes and cohort dependent. But I think 3 to 5 is probably, I don't know, a ballpark number. It's a great question though. And then of course the back end of that is long read sequencing. If we do a T to T assembly of these genomes, what do we find unique? There will be a lot coming out. The discoveries are going to live in the STRs. I mean, here's the long and short is nobody in this room knows how to interpret a repeat variant in a centromere in a case of autism. But, an STR that's expanded that we knew lived in the coding region of the genome that we missed before. I think there will be quite a number of significant results there and with VNTRs and
Oh, really?
and those
Yeah,
things.
that's interesting.
I think there's a lot coming in that
Oh, that's
way.
exciting.
Yeah, it's going to be very exciting.
So, so you think a lot of like missing heritability. So how much of the missing heritability then is from not measuring those? Let's add that all up. Indels are easier to measure, but like the structural variation, these complex, the stuff that you really talked about that you've gone beyond the tip of the iceberg now. You're kind of beginning to get it all. How much of the missing heritability do you? Yeah.
I don't think a major fraction, not to undercut our own funding, but I don't think a major fraction of the missing heritability itself is this being published. No, I think that we've been pretty open about this. And in fact, in Nomad, we went to lengths to show this, that a major fraction of the missing heritability is very unlikely to be explained by structural variants because, again, they live on those haplotypes that are already tagged.
Yeah. And
the average LD with a structural variant in an SNV is about 0.85. I think that what's been more profitable is dissection of the causal variant, where you've already measured the association, but you don't know what's causing it. And I think where we can get to a structural variant, we already have several examples of this in the UK Biobank and in other places. Along those lines, by the way, something I didn't have time to talk about, in the very, very near future, there's already a paper out. We'll be taking the panels of long read sequencing that we have, like in all of us, where we get 25,000 structural variants per individual, and we have about 9 million structural variants, it looks like, in the first 1,000, and imputing those into the short read data. So it looks like we can increase our yields to about 18,000 structural variants per genome by just taking the panel of long read assemblies and imputing those on. So now when you want to start to ask questions about the common variants you have or the haplotypes you're looking at, you'll know exactly what structural variants may be living in that neighborhood. Yeah. And I think that'll be really helpful for a lot of studies.
I have another question, but I wanted to leave it open.
I know I'm a few minutes over, so if anybody needs to
run,
please.
People have their stuff. I'll ask one more thing. I was noticing, but it may be not that really important, but I wanted to get your take on it. It's like in the last part in the fetal diagnostics, and I noticed this in the paper, that the yield from inherited variation from the mother, it's a little harder. It's like the de novos get called very, very clearly. Inherited might be a little bit harder. Rare, like inherited variation. I want to. So how does that?
You mean in the non-invasive?
In the non-invasive,
yeah,
screening, like when
100%. It's actually really hard.
Yeah. So I was thinking about that and going, okay, well, inherited variation, we have an inherited disease, right? It's the heritability is 70 or 80%, so that's kind of a problem. But then I was thinking, what's the purpose of the screening? The purpose of the screening isn't to identify polygenic risk. Well, I'd love to hear you talk about that a little bit, but like my thinking about it is the reason to do this is to identify haploinsufficient genes or big mutations that then you want to eventually correct with gene therapy in utero. And so so this is fine for that, it seems to me, and I want to, just in my
I
thinking.
mean, that's a decade ahead, but that is
Yeah.
exactly what we want to do. Exactly right.
So I'm thinking, but like how, yeah, so how do you take this other piece of this heritable variation? Like do you have ways of thinking about how one can kind of improve?
Yes, we're working very hard on that. So what I said very quickly, I should probably just do a whole talk on this, but is the de novo variance, no matter what the fetal fraction being correlated with how early we can detect these things, is we can find them. Dan very, very cleverly sees this, which is really the thorn in our side right now, which is if the mom has the variant and the proband has the variant, your allele fractions are not separated by very much because you only have this tiny additional allele fraction on the variant. For the inherited variants that cause disease, I think we're okay because that yield is actually quite low, right? In autism and neurodevelopmental disorders, where it's killer is recessives. So known recessives that are interpretable and reportable.
I see.
Very, very hard. So going back to our Bayesian models in autism, we're doing the same thing here, where the one thing I have though is a almost perfect maternal carrier screen. So the question becomes, if I know the mom has a variant and it's valid in the carrier screen, can I now begin to weight variance by the likelihood that it lives in the fetus based on the coverage, the fetal fraction, and the existence in mom, and then begin to pull out the genes of interest? And that's what we're doing. So we're doing a lot of work there. I think we're going to crack this. But I think that conservatively, the intro to this is really saying, let's get rid of amniocentesis for discovery of de novo variants. And that'll solve 80% of the problem.
Yeah,
The 20% is going to have a long tail.
Yeah. Those who had amniocentesis, and I haven't, but I've been very much a part of it. It's an incredibly stressful thing. So it's great. Okay, well, thank you a lot, Mike.
Thank you.
Yeah.